Hello friends, welcome back to another video from Shomu's Biology. If you are a layman, if you want to understand what is Rt Pcr test and how Rt Pcr test is done, what is the procedure of Rt Pcr test and how Rt Pcr test is being used to detect the presence of SARS-CoV-2 or coronavirus in your body, then watch this video to the end. So the very first thing, if you want to understand Rt Pcr test, because I assume that not all of you are from science background, so I want to explain it as simple as possible. I won't explain much technical things, but you need to understand two things very quickly. The very first thing that you need to understand is that any organism in this planet, they have their genetic material, which is actually controlling everything of that organism. Okay, we have this genetic material, and that is DNA in our case. In all the animals that we see, they have this DNA as a genetic material. But in case of the virus that we are talking about, okay, viruses may have DNA or may have RNA as their genetic material. In case of the virus that we are talking about, that is a novel coronavirus, in this case SARS-CoV-2, they have RNA as a genetic material. Okay, SARS-CoV-1 also had the same RNA as a genetic material. Now, what is the difference between this DNA and RNA? Both are nucleic acid. Okay, generally, like you probably heard the name protein, carbohydrates, right, fat, because we eat these things. We also eat nucleic acids in the food, in plant foods, in animal foods, everywhere it's there. But we generally don't talk about that much. It's more of a information that is stored in an organism's body. So that makes that organism who they are really. Okay, so the DNA and RNA, what's the difference is that DNA is double stranded, and on the other hand, if I write RNA is single stranded. What do you mean by this stranded? Means how they look like. Okay, the DNA looks something like a ladder structure like this. Okay, and double strand means there are two sides of the ladder. You can see there are two sides of the ladder, and then this ladder becomes twisted. I haven't drawn the twisted version. The ladder normally becomes twisted. And in case of RNA, it is built with only half of what the DNA is made up with. So as per the structure, DNA is double stranded because it contains two different strands, while RNA contains single strand. We call it single stranded. Now DNA contains deoxyribose sugar. In RNA, only ribose sugar is present. That's the difference between RNA and DNA, okay, in very basic way. Now the thing is, in our body, the DNA is the way the informations are stored inside cell, okay, DNA is the way it's stored. But the DNA needs to be converted into RNA, and the RNA needs to be converted into proteins, and these proteins do everything in our body is done by these proteins. Literally what you can see right now is a build of protein, okay, because literally this is known as an expression. So DNA to RNA, RNA to protein, this is the sequence of how we work, how all these animals and most of the plants, bacteria also, how we work. On the other hand, this virus, they start with RNA, they don't have any DNA, they don't need to deal with the DNA. Okay, now why I'm talking about this? The thing is that DNA can be converted to RNA, RNA can be converted to proteins. Okay, the DNA conversion to the RNA is known as transcription, RNA into protein known as translation. Okay, these are the two different terms. So this is transcription, converting DNA into RNA. This is a normal way, DNA to RNA, RNA to proteins. But in case of this virus, it starts with RNA, so it only makes protein. This is one way this virus can make proteins faster, because there is one step bypass. They don't need to go through transcription, they directly make the proteins. On the other hand, there is a way that we can go back, not from protein to RNA. No, proteins are required to make DNA, proteins are required to make RNA during the process of the synthesis. It's needed for different purpose. But we can go from RNA to DNA. That's the reverse way, backward way. We call it reverse transcription. Okay, this is the one thing that you need to understand. We can convert RNA into DNA, and this is really important, because when we talk about Rt Pcr, this Rt stands for reverse transcriptase PCR, because to go from DNA to RNA we need enzyme. Means again we need protein, specific protein to do that. And the name of the protein here is RNA polymerase. This is the name of the protein. While we need to go from RNA to DNA, we need a protein, and the name of the protein is reverse transcriptase. Short form RT, in this case of Rt Pcr. RT stands for reverse transcriptase. Now what PCR stands for? Let's talk about that. PCR. P for polymerase, C for chain, R for reaction. Polymerase chain reaction. Now what do we mean by polymerase chain reaction? Let's combine reverse transcription with polymerase chain reaction. These are two separate things. We have polymerase chain reaction, we have normal PCR as a diagnostic techniques for many years. Okay, it's there. It's being used as a molecular biology technique as well as in a diagnostic technique. But we combine reverse transcription with PCR, and how we do that? Let's first understand PCR here, okay? What is PCR and how it's done? In PCR, assume there is a DNA. Let me draw the DNA here, okay? There is this DNA. And we know what this DNA is made up of. Generally, the DNA has sequences. What do you mean by sequence? Generally, DNA is composed of A, T, G, C. These 4 letters. They are made up with these 4 letters. Let's assume this is one of such example, a segment of the DNA with this much of letter. Random, I just wrote it, okay? Now we know this sequence. So the sequence is known sequence. We know the sequence. And now what we want to do is that we get this DNA, we want to make more copies of this DNA, just doing like a Xerox machine. Xerox of DNA. Just like, you know, in Xerox place, what you do is just take a paper and say that give me copies of that. No matter how many small the size of the paper is, let's say you bring one paper only and you tell them I need 10 lakh Xerox copies. They will give you 10 lakh. No matter how many copies you want, they can give you that. The same thing happens with PCR. We get a known sequence of DNA and we can make as many copies as we want from that single DNA. Okay. So the process is carried out utilizing a machine known as Thermocycler. Okay. The machine is very simple. It simply changes temperature from time to time. That's it. That's what the machine does. And we need some reaction mix and all. I'm not going to talk about technical about it. But the idea, let's assume we know this sequence, right? And remember one thing, this DNA is designed in such a way that A always pairs with T, T always pairs with A, G pairs with C, C pairs with G. So where we have A in the opposite side, it will be T. Where we have T in opposite, we have A. Where we have G in opposite, we have C. Where we have C in opposite, we have G. A opposite of T. Okay. And now what we want to do, we want to make copy of this DNA. So what we can simply do is that we need to separate these 2 strands from each other, right? As I said, DNA is double stranded. They have 2 strands. So you need to separate the strands from each other. So the thing we need is heat, high heat, 95 degrees Celsius temperature, for example, 90 degree, 95 degrees Celsius temperature. It will separate the strands from each other. 2 strands are now separated. They are not linked together. Normally they are adhered together. A pairs with T with 2 hydrogen bonds, G pairs with C with 3 hydrogen bonds. So these bonds are broken when we have very high temperature, they are separated. Very first step, step number 1. The second step is what is known as probing and simply priming, what we can say. Priming. Now what we mean priming is that we design a small probe. What we mean by probe? In this red color I draw this. Small segment, 15 to 20 nucleotides long. Nucleotide means this A, B, A, T, G, C, these letters. 15 to 20 such letters long. Okay. So we design this as a primer. We call it primer. And we have 1 pair of primer. In this case, 1 pair means 2 primer. 2 primers are used in 2 opposite directions. They will bind to it. How to design the primer? Now if you ask, remember, we know if there is A in the opposite, T should pair with it. If there is G in the opposite, C should pair with it. So if we know that this is a sequence AATT and our primer needs to bind here, the primer sequence will be TTAA, right? So we design the primer, we can design it. Scientists have designed it, it's ready, is in the reaction, we add it and we lower this temperature down a little bit, because if we don't lower the temperature down, this primer cannot attach to the already separated single strand form of the DNA. We call it annealing of the primer. So for that the temperature need to be reduced to 50 to 55 degrees Celsius temperature. It varies, there is a range because it varies depending on what species we are dealing with, what sequence is present there, and so many things are responsible there. But generally this is a 50 to 55, let's say 55, 54 degrees Celsius temperature, lowering down from 95 in order to anneal the primer with the DNA. Once annealing is done, then the step number, so this is the second step. The step number 3 now is polymerization. Polymerization. Polymerization means what will happen now? We will add plain ATGC into the reaction mix. Single letters. So that now whenever there is a G, C will go and bind with it. Whenever there is A, T will go and bind with it. This way they will bind, and this is known as polymerization. It will be catalyzed, I use a lot of scientific terms here. It is actually done by the enzyme, in this case DNA polymerase. Okay, but normal DNA polymerase is not used, a special kind of DNA polymerase is used in this case. So this again, what? It's a protein. Okay, it's a protein. So they continue and they will make the new strands. So as a result, what will happen after the third step? What we will have? We will have 2 copies of the same DNA that we have started with. So it was 1, now we made 2. So from 1 to 2. Remember Xerox, you take one paper, from 1 they can give you 2. Now individual 2 can now give you what? So now let me draw this quickly here and further. So from 1 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32, 32 to 64, 64 to 128, 120 to 256. So it will continue like this. See, it's rapid. So this conversion from 1 to 2, 2 to 4, individual conversion is known as Pcr cycle. They are known as Pcr cycle. So from 1 to 2 cycle number 1, from 2 to 4 cycle number 2, from 4 to 8 cycle number 3, 8 to 16 cycle number 4, 16 to 32 cycle number 5, 32 to 64 cycle number 6. So it will continue like this. Okay, for how long? Just calculate. Using calculator you can also do that. If you are a science student, math student, an engineer, physics student, you know the formula 2 to the power n for those. Okay. So this is how you can calculate. This is a rapid amplification. This is how the amplification is done. Right. This is known as Pcr cycle. Now this cycle is very, very important. Keep this in your mind. Pcr cycle is very important because we find out CT value based on the Pcr cycle. Because probably you have heard this term CT value. I have seen this in newspapers and places. So what is CT value? We'll talk about that as well. This is known as cycle threshold value. We'll talk about that later. So now this is a normal PCR process that we amplify. But what is Rt Pcr? Real time. The Rt Pcr works in real time manner. So in many cases we also say it's real time PCR. Reverse transcriptase PCR. In both the case we use this term Rt Pcr. So the Rt Pcr that we are utilizing here as a term, many people may comment from the first place that Rt means in this case it can be real time PCR. In this case we are using real time PCR, we are using reverse transcriptase PCR. So in both those cases the short form is Rt Pcr. This is also known as QRT PCR, small q. Q means quantitative real time PCR. So what happens in real time PCR is that the moment one DNA becomes 2, 2 becomes 4, 4 become 8, the signal is something there. The signal is in terms of light intensity that a machine can recognize, and based on the intensity of the light that we can identify how many cycles have completed, how much quantity of the DNA is replicated and made into newer copies that we can get from that real-time manner. Okay, that is why we call it real-time PCR. And why reverse transcriptase? Because imagine in SARS-CoV-2 the novel coronavirus carries a RNA as a genetic material, and in this case I taught you the process of PCR with DNA, and PCR always works with DNA. So if we need to amplify the viral RNA cannot be amplified. We need to convert that RNA into DNA first, and then that DNA can be amplified. So the viral RNA first is converted to the DNA, and conversion of RNA into DNA is made by the enzyme reverse transcriptase through the process known as reverse transcription. Reverse transcription. So reverse transcription technology is used converting viral RNA into DNA first, and then that viral DNA which is formed here will be amplified with different PCR cycle until we get the number of load or number of the viral DNA that we want. Now you may ask, what is the importance of this and why we are using as a test? We are using Rt Pcr as a confirmatory test for coronavirus infection, because let's say in your body there is very little amount of coronavirus present, so very little amount of RNA is present. So what we need to do, we need to take the sample and we need to amplify the RNA to check, because in order to check we need quantifiable amounts. The RNA is so less in number, so small in size that we cannot see it with even light microscope. RNA DNA cannot be visualized with microscope like that, RNA particularly. DNA in a form chromosome can be visualized, but that's a separate idea. We need specific amount, we need a bigger amount of RNA in order to work with it, in order to know whether the virus is present in our body, and Rt Pcr test help us to find that. Okay. So in Rt Pcr there is only one extra step, viral RNA converted to DNA using the enzyme reverse transcriptase, and then rest of the process are similar that I have already discussed. So now what is this CT value? CT full form, remember I told you, cycle threshold. What is cycle threshold? Cycle threshold is a value that tells us after how many cycles the load of viral component of viral genetic component, that is in this case RNA, in case of PCR it's DNA, after how many cycles the total genetic material of the virus is detectable, that is counted with CT value. Okay, adequate amount formation or conversion of one single or two single, whatever amount is present in your body, into an adequate amount is necessary in order to detect it. So how many cycles is necessary to detect the quantifiable amount of the viral genetic material in your body? That is the CT value. Now the CT value, now imagine one thing, there is Ram and there is Shyam. Okay, both they gave samples and we run the Rt Pcr, and what we found out is that in case of Ram the CT value was 20, in case of Shyam the CT value of 36. What does that mean? That means in case of Ram we need to go for 20 rounds of PCR, 20 rounds of Rt Pcr to properly detect the adequate quantity to call it as a serious infection. Okay, we made a level, that this is a level, means a particular concentration of this viral genetic material. Once this level is reached, we call it as a serious infection, and in our case the level is set to 35. So if the CT value of 35, if someone has a CT value of less than 35, that means the person is COVID positive, because in case of Ram, as I said, 20 cycles are needed. That means that that person Ram already had higher load of virus in his body, right? Try to understand, try to correlate. 20 cycles only needed to get the same amount of viral load compared to the Shyam, because Shyam needed 36 PCR cycles to reach the same amount of the viral load. That means originally in the body of sham, the concentration of the viral load is less, while in case of ram, the viral load was more. So in our case, 35 is set as a point. So if the CT value is less than 35, the person is COVID positive. The CT value more than 35, the person is COVID negative. There is nothing much difference about other, like you don't need to know exact value of exact CT value in your body, because CT value just gives us an idea whether you call a person seriously infected or not. You call it a person infected or not, right? Positive or negative. Apart from that, it does not provide us any information, because let's assume in this case what I said, that in case of ram sham, so in case of sham we have 36 of CT value, in case of ram we have 20 CT value. So as per this value we can say that yeah, ram is more affected by this virus than sham. True. Now let's compare ram with another person, ram with X. Now ram has a CT value of 20, X let's say has a CT value of 25. So comparing both, what you can tell, that yes, ram is more infected, sham X is less infected. But no, you cannot comment on how the infection is there. You can comment that yes, ram has more load of virus initially than X, but that does not necessarily mean the ram clinically will be more symptomatic than X. It may happen that X is more, the effect in the X's body is more severe than the effect of coronavirus in ram's body, because how our body and how our immune system will react against the viral load, against the viral infection, is not determined by the CT value. So you don't need to know the CT value, just need to know whether you are above 35 or below 35. Below 35 you are infected, above 35 we give a clean sheet that yes, it's past that area. But still, even if it's above 35, and even a little amount of the RNA of this novel coronavirus is present in your body, it's detected, you will be tagged as a positive. You will be tagged as a positive, because this is a highly sensitive test, highly accurate test that will give you a data. Because imagine why it's highly accurate, because now you'll understand many people are spreading false propaganda about the Rt Pcr test. The thing is, in Rt Pcr we designed this primer, what we can say, primer very carefully. And as I told you that A always pair with T, G pairs with C, and there is no other way, there is no other rule. So it's just like Velcro, perfect fit. So if the primer does not bind with the target, then fit will not be there, and if there is no fit, there is no indication. If there is no fit, there is no indication. Okay, this is really important, you need to understand. So if you get, even if you have a tiny amount of match with novel coronavirus RNA present in your body, it's going to be detected. There is no chance. False is very rare. False is very rare. Yes, false negative is possible due to the sampling error, but false positive is very rare in case of Rt Pcr. Now I told you an overall idea about PCR. I told you what is CT value and how we measure CT value and why CT value is important, where we cannot rely on CT value. Now I am to talk about the exact mechanism of this Rt Pcr. As I told you earlier, that the Rt Pcr process is real-time mode of detecting the amplification of the DNA. First convert the RNA into DNA and then convert the DNA, copy the DNA into multiple forms. The very first step is conversion of RNA into DNA, and then DNA is multiplied. But the question is how exactly the machine recognizes the DNA, how we can quantify the DNA, how the quantification is done. As I said, the quantification will be done in real time. Okay, so how it's done, let's talk about it. The quantification is done in a way that we know as either we can use a probe or we can use a dye. Now the probe can be fluorescence type or it can be hybridization type. There are these two different types of probe. One uses a technology known as FRET, the other one uses hybridization technology. Now what we mean by this, let's talk about that. If you want to know about the details, then you can go, otherwise you can skip. If you understand, then please stay and listen to this properly. So here it is, let's say this is the DNA and this is our primer which is already added, the primer is added here. Okay, the primer is added. There is upstream of whatever it is, and now the new strands being synthesized. And what happens actually, what we use here is, let's say in this case we are using probe. There is one example of probe known as Tachman. Okay. Now what happens in this Tachman probe is that it contains two components. Now I'll be going a little technical, not much. One is this known as fluorescence indicator or fluorophore. Another one is quencher. So I'll tag fluorophore as F and quencher as Q. So this is known as a probe. Okay, the probe also has this letter so that it can pair with the target DNA. This is the Tachman probe. So it has two units, fluorophore and quencher. Fluorophore means emits light. Once receive light from outside, they emit light from itself. Okay, and they are tagged with quencher. Quencher is another component that prevents the release of light from the fluorophore. So once this quencher is attached with the fluorophore with this DNA in the middle, the fluorescence is prevented. No fluorescence, no light emission. No light emission. Okay. Now what happens, the moment this new DNA is being synthesized, like this, and the more they move forward, they are going to cut the link between the fluorophore and quencher, and they will replace them, and they will replace it and fill with the new DNA. So whenever they cut the fluorophore from quencher, so now I can draw this, the fluorophore is free, the quencher is separated. So this fluorophore now can emit light. This we can measure. Light intensity we can measure and we can detect. The higher the intensity, we can tell the more DNA is being amplified with Rt Pcr. Got it? So after each cycle, this light intensity increases. Fine. This is when we use a probe. In many cases, in this case of Rt Pcr, mostly we are using dye. And the dye, one example, cyber green is an example of dye. It's more easier. This dye only binds to double stranded form, never binds to single stranded form. Okay. So imagine in the very first step, whenever the DNA strands are separated from each other, and in that same mix we put this dye, cyber green, but the dye is not binding to the DNA because they don't bind to single stranded DNA. So from this sample we get light, we get intensity, but very low, because only the dye which is present in the liquid, in the suspension, gives us a little amount, is not much. But whenever the separated and new strands being synthesized, new strand is added, then what will happen? Let me draw here. Okay, new strands added, double stranded DNA created, then this dye binds to it and gives us light, lots of light intensity which can be measured. So whenever we increase the number of DNA, amplify the number of DNA, the light intensity increases. So this is the way we measure the quantity of amplified DNA in the sample. So we take the sample, a nasal swab, it's taken, and then we put it into a mixture of chemicals, and with this nucleotides A, T, G, C, all these things are provided, primer is provided, we put them into the reaction mix, and the process works automatically and gives us data. That's how it works. Now you may ask question, many people have asked this question earlier, that when the new coronavirus strains are coming, there is a mutation in coronavirus strains, can this test detect it? Yes, why not? It can detect any DNA sequence if we know the sequence itself. So whatever new form of the coronavirus are coming in, the very first thing that researchers need to do is to sequence it. Sequencing a DNA means knowing what the, in case of coronavirus, sequencing RNA means knowing what exact nucleotides are present in the RNA. Once you know that, we can design primer according to it. Once we design primer, we will add that primer and the reaction will work like a charm. Okay, so this is the process and there is no doubt about it. This is one of the best techniques. This is the best techniques that is available right now to detect RNA and DNA in a sample. Earlier there are techniques known as southern blotting and northern blotting. Southern blotting was there to detect the presence of DNA, northern blotting was there to presence the detect of RNA, but those were really complicated, a lot of time consuming. But now RT-PCR can be done very rapidly, fast, and in a single run frame, time frame, we can run as many as 96 and even more than that some cases run in RT-PCR machine. There are different wells and systems are there, so based on that we can run RT-PCR test a lot with these machines, with the Thermocycler machine. So this is the way RT-PCR test is used for detecting coronavirus. So RT-PCR, whether you can call it real-time PCR, better to call it qRT-PCR, means quantitative real-time PCR, and you can also call it as a reverse transcriptase PCR. But yes, the data is got in the real-time manner and the process utilizes reverse transcriptase enzyme. So that's all about RT-PCR test for COVID-19 and COVID-19 test with RT-PCR. If you like this video, please hit the like button, share this video with your friends and colleagues and subscribe to this channel to get more videos like that. And if you don't understand English, if you want this video in Hindi, then you can watch this video in Hindi version also in my channel. Thank you.